This is Chapter 20, Eukaryotic Diversity Review. I will review some of the brief features of eukaryotic microorganisms, including phylogeny of eukaryotes. We are going to focus on fungi, some of the key distinctions between primary and secondary algae, features of amoeba, alveolates, and some of the parasitic protozoa, because there are several protists causing serious disease to humans. In this chapter, you don't have to remember the details about eukaryotic microorganisms, as there are so much information provided in this slide. So I'm going to point out the key features that you need to at least know to do better in the final exam. As you may already know, all the eukaryotic system are quite diverse, and from the individual single-cellular microorganisms to complex multicellular animals, they are quite diverse, and they have so many different groups evolved among eukaryotic system. This is the eukaryotic phylogeny. You may want to focus on Opisthokonts, which include animal, fungi, and microsporidia. So you might be surprised that fungi is actually closely related to animals. Another group that you want to focus is Viridiplantae, which include plant and primary algae. So all of these included in Viridiplantae are phototrophic. Here two groups are included in Amebas, which contains Amebas that we know that it has pseudopod, slime mold, and some of the interesting amoeba strains forming filamentous pseudopods are included in these two protistic groups. We'll also review Alveolata, which contains the ciliate or flagellated structure, and also Heterokonta, which are the protists with dissimilar flagella. Especially in Heterokonta, it contains several phototrophic organisms. Those are also related to marine environment. This slide is a summary of each eukaryotic groups. We will go on to a very brief introduction to each group on the next following slides. Opisthokonts include animals and fungi. Term Opisthokonts indicating backward spear, which means a cell with one single tail. Fungi and multicellular animals both have a stage with a singular tail. In the case of animals, sperm. Some of the fungal species also have spores, has a motility. Not only the feature, but also genetically, animals and fungi are quite related, because both animals and fungi share key gene similarity, especially on those genes governing the cellular function, like protein translation, has similar sequence insertion on a certain protein sequence. For example, EF1 alpha, which is the elongation factor alpha of translation. Both animals and fungi has 12 amino acid sequence insertion. As you can see on this table, shown in the yellow box are Opisthokonts, animal, fungi, and microsporidians. All of them has 12 amino acid insertion sequence shown here, which is not found in either plant or other protistic organisms. Flagellate is the most closely related protist to animal. So far, the evidence pointed that Flagellates have diverged from animals about 600 million years ago. And genetic studies also have shown that several genes found in animals also found in Flagellates. Moreover, there is a similarity of the feature between Flagellate and choanocyte, which is part of the sponge body. As you can see here, this is the Flagellate nucleus. It has color and single flagellum. You can also see the choanocyte, which has a head and the flagellum together. So scientists assume that probably individual choanoflagellates got together to form multicellular organisms. Viridae plantae include plant and algae. Both plant and algae acquire phototrophic microorganisms and were able to do photoautotrophy. Depends on how the chloroplast is internalized and how it stays in the inside of the cells. The Viridae plantae is separated into two major groups. One is primary endosymbionts. Second one is the secondary endosymbionts. Primary endosymbionts include green algae and red algae. Secondary endosymbionts include cryptophyte algae and chrysophytes, which is the kelp and diatoms. This is the structure of the green algae and red algae. Both of them are included in primary algae. As you can see here, the primary host nucleus and the chloroplast are both inside of the one single membrane, which is pretty similar to plant cell. However, the secondary algae has a little more separated structure than the primary algae. As you can see, this is the original nucleus, and this is the additional membrane capturing the chloroplast and the nucleus, probably from the algae that this large cell has engulfed. So the secondary algae looks like it's not completely merged endosymbiosis of the chloroplast or photosynthetic microorganisms with the large cell. Other than Opisthokonts and Viridae plantae, there are several diverged groups included in Protists. Protists include several distantly related eukaryotes, so those are grouped based on their morphology, lifestyle, and their genetic features. So three large groups that you want to remember is Amebas, Alveolates, Heterokonts. Eugulenozoa and Excavata is a quite distanced group which has shown extensive evolutionary reduction. So some of them lost mitochondria, some of them has partially functional one, etc. Some of the protists included in these groups are also related to human disease, so we are going to focus on those areas as well. I'll skip this. So let's start with fungi. Fungi play a significant role in ecosystem. We reviewed briefly in chapter 21, or you guys probably have seen the role of fungi in symbiosis while you were doing home assignment. Fungi provide a essential support for all communities of multicellular organisms. Especially fungi is the major decomposer, so it plays a significant role in recycling the biomass. Also, fungi has an important role in ecosystem as a mutualistic symbiont of plant. We reviewed briefly about mycorrhizae symbiosis in chapter 21, which is the extensive mutualistic symbiosis between plant and the fungi. About 90 or more % of plants existing on Earth has a mycorrhizal relationship, which get benefit from the absorbed nutrient by fungi. These are some of the positive aspects of fungi, but fungi are also a part of the large pathogenic groups, especially to plants. Recently, there are more reports suggesting fungi could be as life-threatening opportunistic pathogens in case the humans with immunocompromised conditions. Also, some fungi are pretty important for the industrial purposes, like producing antibiotics, producing enzymes, producing food products such as wine and cheese. The shared traits of most of the fungal species is one, it has absorptive nutrition. Fungi actually secrete enzymes and break down the polymer structures and absorb those broken pieces for their nutrient acquisition. Also, fungi have a distinctive elongated mycelial structure. So it begins with hyphal formation And once hyphae elongates and form branched hyphae, eventually it grows as a mycelium, which is much bigger structure. Another distinctive feature of fungi is that fungi has cell walls. So there are two groups of eukaryotic system have cell walls. One is plant, the second one is fungi. Plant cell wall is made up with cellulose. The fungal cell wall is made up with glucan, and chitin is the distinctive component of the cell wall, and also it contains mannose. Fungal membrane looks pretty similar to mammalian system, but it contains ergosterol instead of cholesterol found in animal or plant. So this is the mycelium structure. This is the chitin. Individual subunit of chitin is N-acetylglucosamine, which is the sugar derivatives. And acetylglucosamine is also part of bacterial peptidoglycan polymer. Fungi has not only the hyphal cells, but also some fungi form unicellular structure, which we call yeast. Yeast is reproducing by budding. Basically, the small bud grow at one side of the mother cell and grow much bigger, eventually cut and become two individual cells. These are the bud scars showing where the old daughter cells separated from the mother cell. So Saccharomyces cerevisiae and Candida albicans are two important microorganisms impact human life. Saccharomyces cerevisiae is a baker's yeast. Candida albicans has been normal flora and now become opportunistic pathogens among the immunocompromised population. Also, Candida albicans is the major cause of yeast infection. There are fungi with mycelial form or forming mushrooms or having mycorrhizal relationship to plant. So based on their lifestyle and sexual cycle, there are 4 major clades included in fungi: Chytridiomycota, Zygomycota, Ascomycota, Basidiomycota. You don't need to know individual fungi under this group, but at least you need to know those key features of each category. The key feature of Chytridiomycota is that this is the most deepest branching clade of fungi, which means that the earliest branched out group of fungal species. The key thing that you need to know about Chytridiomycota is that these fungi produce motile zoospore. So this is one of the evidence why fungi is included in Opisthokonts. Chytrid is a symbiotic partner of kettles, breaking down fibers. Also, there are chytrid species causing serious infection on the freshwater frog species. They impact significantly on the extinction of frog species in Latin America. Zygomycota include the organisms that you review in the laboratory class. One of them was a rhizopus that you saw, which form asexual spore called sporangia on the structure called sporangium. So this black round circular shape is a sporangium. Instead of the sporangium, there are individual spores are formed. Ascomycota include several groups that are quite familiar to our life. It includes Neurospora, which is a bread mold, morels and truffles. Aspergillus, you also saw this fungi in the laboratory. You probably saw conidiophore, which is the asexual sporulation structure. An individual spore formed at conidiophore is called conidia. Basidiomycota includes several mushrooms that we see quite commonly. Glomeromycota is the group of fungi. All of them included in this group are obligate mutualists of plants. So these are the group of fungi forming a mycorrhizal relationship to humans. Let's move to algae. Algae is the primary producer in all ecosystems, especially in aquatic or marine environment. Algae and the photosynthetic bacteria are known as phytoplankton. As I mentioned earlier, the algae possess chloroplast, but depends on how the chloroplast is associated with the algae cells. One is become primary endosymbiont, the other one is become the secondary endosymbiont. Primary endosymbiotic algae include two major clades, Chlorophyta and Rhodophyta. Chlorophyta are also called green algae. Rhodophyta are also called red algae. Those are dependent on the UV length of the light that they absorb, and then they have different chlorophylls absorbing particular wavelength of the light. A lot of the green algae are unicellular, but there are some algae that's forming multicellular structures. Especially the green algae species contains either single form, multiple cells together, or even form a large structure with the offsprings inside, which mimics the gradual increase the number of cells and forming from single cell to multicellular organisms. So this is a good model for explaining the evolution of multicellularity. We reviewed this in the previous slides that the primary algae has chloroplast, which is quite closely merged to the cell outside. Red algae are colored by the photopigment phycoerythrin. So with this phycoerythrin absorbing longer wavelength, it allows growth in deeper water. Secondary algae arose from ancestral protists engulfed a primary endosymbiotic algae. So chloroplasts of secondary algae are surrounded by more than two membranes. Their metabolism includes heterotrophy. One example of the secondary algae is diatom. Diatom is a unicellular algae, and it has very unique shell called frostole, which is the shell made with silica. So these pretty structures and shapes are from the shell structure of diatoms. Amoeba and slime molds. Amoeba is amorphous form of microorganisms. The amoeba species are free-living predators in soil or water, and amoeba engulfing the prey by phagocytosis. Some of the free-living amoebas are pathogenic, and oftentimes they harbor bacterial pathogens. Slime mold is also included in amoebas, but it has very complex differentiated fruiting bodies that look similar to fungal species. So slime molds were originally grouped in fungi, but later there are quite distinctions in the genetic features, and it's now included as part of amoebas. So this is the distinctive fruiting body structure of slime mold. Alveolates are named for the vacuoles that they possess. The vacuoles are flattened structures, and those vacuoles are called alveoli, and those are found within their outer cortex. So the protists containing alveoli are included in this group. So this group contains quite diverse morphologies: Ciliates, dinoflagellates, and Apicomplexans. Ciliates are the organisms containing large numbers of cilia, which is a thin, short filamentous structure with microtubule network structures. Cilia has two functions. One is for cell propulsion, second is for acquiring food. The most famous ciliates is Paramecium that we use in the laboratory. We probably saw this organism in general microbiology laboratory. This is the morphology of Paramecium, and these are the cilia formed with microtubules. So the second group in alveolates is dinoflagellate. Dinoflagellates are the major group of marine phytoplankton, have two long flagella, and also they have chloroplast, which is surrounded by three membranes. So dinoflagellate is the major cause of red tide with a eutrophication, and dinoflagellate produce toxins poisoning the shellfishes. Apicomplexans usually have distinctive structure, and they have gone through extensive reductive evolution, so it has quite distinctive feature. This is the major group of animal parasite. Cryptosporidium and Toxoplasma are the common disease caused by Apicomplexans. So Apicomplexan has two important structures. One is apical complex, which allows the cells enter into the host cells. Apicoplast, which is the structure function for fatty acid metabolism. This is the causative organism for malaria. Plasmodium is carried by mosquitoes. It has different stages of life cycle in red blood cells. So if you want to know more about the life cycle of the Plasmodium, we have nice animation, so you can take a look at this. Now we are going to review some of the parasitic protozoa species. Especially humans' intestine has many kinds of protozoa. Most of them are harmless, and they inhabit in human intestine as commensal organisms. But there are several protozoans that are parasitic and may cause deadly illness. So one of the parasitic protozoan species is Trypanosoma. Trypanosomes belongs to the group Euglenidae, and they are obligate parasites. Trypanosomes cause serious illness, especially in Africa. Leishmania major is the cause of leishmaniasis. Trypanosoma brucei cause African sleeping sickness, and this is the trypanosoma in the blood cell. Metamonads also including several important parasitic groups. One of them is Giardia species. Giardia species is an intestinal parasite. It lacks mitochondria, and it has life cycle alternating between two major forms, trophozoite and dormant cyst. So this is the major cause of waterborne illness. If you happen to be in the countries or area where there is not pure water available, then the water oftentimes contaminated with the Giardia species cause intestinal disease. So this is the microscopic image of Giardia species. Has multiple flagella, and it can go through a life cycle. It infects epithelium of the intestine. Other intestinal parasites include amoeba, Apicomplexans and Cryptosporidium, Balantium, and some of the Microsporidian species. So you don't need to know about these, but at least you should know about Plasmodium falciparum, which is the cause of malaria, and also Trypanosoma and Giardia.